We'll take a look at the prokaryotic cell membrane and membrane transport in this lecture. There are lots of structures that are internal to the cell wall in bacterial cells, and right inside the cell wall we have the plasma membrane, which we're going to focus on in this lecture, and then the rest of this we'll talk about later. The bacterial plasma membrane is very similar to the plasma membrane in eukaryotic cells. It is made of a phospholipid bilayer, which has proteins and carbohydrates in it. The functions of the bacterial plasma membrane are very similar to the function of the plasma membrane in eukaryotic cells. So it is selectively permeable. It's a barrier that's going to separate what's inside the cell from what's outside. And being selectively permeable means that the membrane is going to control what goes in and out of the cell. So there are some things like hydrophobic molecules in water that are able to diffuse right through the membrane, but other hydrophilic and larger polar molecules and ions are not able to cross the membrane. So they need to be transported across with a transport protein. And then what's really different about the bacterial plasma membrane from eukaryotic cells is that because the bacterial cells don't have organelles, they do need a place to do some of the same things like making ATP and photosynthesis as what happens in the eukaryotic organelles. And so they're just going to do those things in the plasma membrane instead, since they don't have things like mitochondria and chloroplasts. So when they make ATP, and if they're using aerobic or anaerobic respiration, they have an electron transport chain. And that electron transport chain is actually just embedded right in the plasma membrane because they don't have mitochondria to put the electron transport chain in. Photosynthesis is the same idea. Because there's no chloroplasts to put the photosystems to gather the sunlight and do the reactions of the light-dependent reactions, we're going to put those photosystems and electron transport chains right in the plasma membrane as well. And then also they do have some enzymes in their plasma membrane that will break down some of the nutrients into smaller pieces so then they can fit through their transport proteins and get them inside the cell. Some bacteria will have infoldings of their plasma membrane, so they will just fold it to the inside. And on the cyanobacteria on the right, for example, you can see all these infoldings of the plasma membrane. And remember, cyanobacteria are photosynthetic, so this is where they're going to put their photosystems and electron transport chains to do photosynthesis. And what's really interesting about these infoldings of the plasma membrane, you can see how maybe these were precursors to developing organelles, or at least some of the organelles. The structure of the bacterial plasma membrane is very similar to what we see in eukaryotic cells. So we've got the phospholipid bilayer. We do have some proteins in the membrane. They do have some peripheral membrane proteins, which remember those are just loosely connected to the inside of the membrane. That's this purple protein over here on the right. And then we do have the integral membrane proteins. So they're going to be embedded within the phospholipid bilayer. And they could be doing a lot of different functions. They might be transport proteins. Maybe they're part of an electron transport chain, which is being used to make ATP and aerobic respiration, or maybe it's a photosystem, part of that to do photosynthesis. Maybe it's an enzyme to take nutrients, chop them into smaller pieces so then they can fit through the transport proteins. So a lot of different functions for those integral proteins. And then we also have some carbohydrates in the membrane as well. And the carbohydrates will be attached to either the proteins or the phospholipids, making glycoproteins and glycolipids. We use the fluid mosaic model to describe the structure of the plasma membrane. And this is just like you learned with the eukaryotic cells. So remember that fluid refers to the fact that the phospholipids are moving. So it's not a static structure. So it will move, and the proteins in the membrane can move around. So it is a fluid structure and not, something that's just solid and doesn't move at all. Mosaic is referring to the fact that there are lots of different types of proteins that are in the membrane, and we have some different carbohydrates. So there's lots of different pieces that are embedded in the membrane, and that's what mosaic is referring to. So like a mosaic picture made out of different colored tiles, it's the same idea here. We describe the plasma membrane as being a mosaic because there's lots of different proteins and carbohydrates that are in the membrane. There's lots of different ways to move molecules into and out of the bacterial cells. So we're going to be talking about all of these different transport methods. And it's good to know which ones are active and which are passive. So remember, passive transport does not require energy because molecules are going down their gradient, going from high to low concentration. So the ones that are passive are the first three. And if you remember, diffusion, anytime you see that word diffusion, that's always passive. You're always going down the gradient. So we have simple diffusion and facilitated diffusion. And then osmosis is the diffusion of water, that's also passive. And then the last four, these are all active. They all require energy. Okay, and there are some differences between the eukaryotes and prokaryotes as far as who does which type of membrane transport. All of them will do simple diffusion, facilitated diffusion, osmosis, and primary and secondary active transport. But group translocation, that's new. You haven't learned about that before. That's only done in prokaryotic cells. So just the bacteria and archaea are able to do group translocation. And then for exocytosis and endocytosis, eukaryotes can do both of those. But exocytosis can only be done by some gram-negative bacteria, and otherwise bacteria are not going to be doing exocytosis or endocytosis. And it's really important to remember which things can cross the membrane and which things can't. So we'll just kind of make a list here. So can diffuse through the membrane without any transport protein. So remember the phospholipid bilayer. The fatty acid tails will point towards the middle because they don't like to be around the water. And so this middle part of the plasma membrane, where the fatty acid tails, that's hydrophobic. And that's what makes the membrane selectively permeable. So that's going to stop hydrophilic things from going through, except if they're really small, like water can slip through, but otherwise they're going to get stopped. But if you have a hydrophobic molecule, something that is nonpolar, so these would be, for example, the gases like oxygen and carbon dioxide, also our lipids. They're going to be able to just diffuse right through the membrane. And then the exception to this rule would be small polar molecules, which the example there would be water. So the reason water can diffuse through the membrane is that as the phospholipids are moving, they create little gaps between them, and the water can sneak through. And then we have things that cannot diffuse through the membrane. So things that are going to get stuck and stopped by that hydrophobic middle would be your larger polar molecules, which are hydrophilic. Oops. So these would be things like glucose and amino acids, and then ions. So like sodium ions, chloride ions, they're also going to get stopped. So these things are all hydrophilic, and so they're not going to be able to cross the membrane unless there's a transport protein to get them across. All right, so simple diffusion, our first method of membrane transport. It's passive because molecules are going to go from high to low concentrations. So you're going to be going down the concentration gradient, and you don't need any energy to do that. The type of molecules that can move through the membrane by simple diffusion, there's no kind of transport protein used. These would be things that can get across that hydrophobic middle of the membrane. So those hydrophobic nonpolar molecules like oxygen, carbon dioxide, lipids, those can cross by simple diffusion. And then our exception, our small polar molecules like water, can also do simple diffusion. So in simple diffusion, the molecules are just going to move from high to low concentrations. So this picture on the left, you can see that we've got higher concentration up here on top, and we pretty much have no concentration on the bottom. And so these molecules are just going to move right through the membrane and keep doing that until the concentrations are equal on both sides. So that's simple diffusion. So these hydrophobic nonpolar molecules can just go right through the membrane, and small polar molecules like water as well. We'll do simple diffusion. Facilitated diffusion, facilitate means to help. So we're going to be using a carrier protein or channel protein to move molecules across. So things that are going to use a transport protein like these would be things that are hydrophilic, and these would be your larger polar molecules, so like glucose, amino acids, and then your ions as well. Oops. And if you look at the concentration gradient, you can see on this top picture we've got higher concentration outside the cell than inside. And you can see that we're going from high to low concentration through this channel protein that has a little channel through it. So that makes it passive. So we're going to be going down the gradient. And you can see with this carrier protein that's on the bottom picture, the carrier proteins will flip and change shape as they go across the membrane. They're very specific for what they will bind to. And when, for example, maybe this is a glucose carrier protein, when glucose binds, it will change the shape of the carrier protein and flips it to the other side. And you can also see in this animation as well, we've got a carrier protein here changing shape, and we're going from high to low concentration, so passive, no energy needed. And then if we look at this graph comparing simple and facilitated diffusion, it's just showing that the concentration gradient, the larger it gets, the more molecules you can get across the membrane. So with simple diffusion, you can just keep getting more and more molecules across as your concentration gradient gets higher and higher. But with facilitated diffusion, notice that we level off up here at the top. Okay, so as you increase your gradient more and more, you can't get any more across any faster. Okay, and so what we see in facilitated diffusion is saturation. So all the carrier proteins get full, and if you're adding more and more molecules on the one side, you can't get them across any faster. So anytime you're using any kind of a transport protein, you can see saturation. Okay, in simple diffusion, you'll never see saturation, you'll never see flattening like that because there's no transport protein used. Okay, osmosis, that's the diffusion of water across a selectively permeable membrane. So water can cross the membrane, but the solute can't. It's going to be stuck on whatever side it is. Because remember, the solutes, things that dissolve in water, are hydrophilic, and they're not going to cross the membrane unless there's a transport protein. Osmosis is passive, no energy needed, and that's because water is going to move down its concentration gradient, going from high to low water concentration. Sometimes they describe osmosis as moving from low to high solute concentration, and if you look at that low to high, you think it's active, but it's really not. How you can think about this is if you've got a membrane separating two sides here, and let's say we put our solute, maybe we've got sugar, And maybe we put 10 grams of sugar on one side, and the other side we only put 1 gram of sugar. And if you fill up to this 100 ml mark maybe with water, you can see that this left side, oops, is not going to have as much water in it as the other side. And that's because this left side has more solute. You've got more sugar over there. And then you can see this other side has less sugar, lower solute, and so you have more room for water, so the water concentration is higher. And so with osmosis, water would move down its concentration gradient, going from high to low concentration of water. And then you can see why they say water moves from low to high solute, but just look at the water concentration gradient and that'll keep it straight. Water can diffuse through the plasma membrane on its own, but that's kind of a slow process because you got to wait for the little gaps to form in the phospholipid bilayer. So most of the cells will have aquaporins, little water channels, and that's going to move the water across much faster. Okay, osmosis also will affect the cell size with the water moving from one side to the next. And if you were to place a bacterial cell into an isotonic solution, iso means the same or equal, and what they're talking about is that you have the same number of solute particles inside the cell as you do outside. Okay, and so there's not going to be no net movement of water one direction or the other, and so the cell's not going to change in size. But if you throw the bacterial cell into a hypotonic solution, so that just means the solution outside the cell, hypo means less than. You just have less solute particles outside the cell than you do inside. And so another way to think about osmosis is water always wants to go where the solutes are. So water's always going to go where there's more solutes. So if you have less solute out here, you're going to have more water. And inside the cell you have more solute and less water. So water is going to follow those solutes and water will move into the cell. And as water moves into the bacterial cell will swell. And that's why they have a cell wall. Okay, that'll prevent the water from moving in too much and prevent the cell from bursting. But if the cell wall is damaged, then the cell will burst, and that's called osmotic lysis. And then if we put our cell into a hypertonic solution, that means that we have more solute outside the cell than we do inside the cell. So hyper meaning more than. And again, water's going to go where there's more solutes. Okay, so we have less solute inside the cell than we do outside. And that means we've got more water inside than we do outside. And so water is just going to move down its concentration gradient, follow the solutes, and move out of the cell. And what happens to the bacterial cell size is going to shrink. And if enough water moves out, we can get plasmolysis. So as the water moves out and the cell shrinks, the plasma membrane pulls away from the cell wall and it rips the cell membrane, and that will kill the bacteria.